At the last lecture, we finished with this synthesis type of slide that walked through all the different bits and pieces of DNA replication in prokaryotes and eukaryotes that surmounted all of the challenges that I laid out at the beginning of the lecture related to the things that DNA polymerase can do compared to the things that it can't, yet are instrumental for DNA replication to occur. I left out one, which is where we'll begin today. And this speaks to one problem that specifically relates to DNA replication on linear chromosomes, like our linear chromosomes. And it relates to how the ends of those linear chromosomes are effectively replicated and retained. I want to show a cartoon that illustrates what the end replication problem is, and then talk about what biology's solution is for it. In green, we have a linear chromosome zoomed in, or zoomed way out so that you can see the two ends. If we take one of these strands, all right, so 5' to 3', and then we're going to replicate in the anti-parallel direction. Primase comes down, and we'll lay down RNA primer on that free end, have the RNA primer, and then DNA polymerase will elongate through there. But recall what is supposed to happen to those RNA primers. DNA Pol 1, Pol alpha is supposed to come in with its 5' to 3' exonuclease activity and displace those RNA nucleotides and lay down DNA. But that requires it being upstream, so to speak, of the RNA primer to be able to displace the RNA. There is no upstream here. So the DNA polymerase can't bind anywhere and elongate anything to be able to hydrolyze the RNA primer. So on the next cycle, now you don't have that end there, all right? Because the RNA won't be preserved, and you haven't replicated it. So you've whittled away the end of that linear chromosome. All right, now it's 5' to 3'. You lay down the RNA primer in the other direction, elongate, and now you've whittled that end away. The ends of linear chromosomes are called telomeres, and they're called telomeres because they have a particular character in their DNA sequence, and they're also called telomeres because there's an accompanying enzyme that helps resolve or at least maintain this end replication problem. So the ends are called telomeres. The enzyme that works, yes, question? The whole end, yeah, what precisely refers to the telomeres? Most formally, it relates to this repetitive DNA sequence that's characteristic that I have on this slide here. Let's call it any portion of the end that could be pruned down by this end replication problem. So it has an element of a moving target to it, and at the end of this slide, I'll speak to what happens if you pruned it all the way down too much. Okay. Telomerase. Telomerase is also a DNA polymerase. It is a special DNA polymerase because it depends on RNA as the template that is used for DNA synthesis, and that RNA template is pre-bound to the enzyme itself. And I'll walk through the cartoon on the right-hand side. I spoke already briefly about if you will, a consensus sequence for what telomeres are. These are called telomere repeats, and it's this repetitive sequence of G's and T's that are shown in the cartoon here. So that GT rich with the repeating pattern there, that's the telomere repeat. And young cells that haven't replicated very much should have pretty long repeats of those G's and T's. The presence of the G's and T's does not solve the end replication problem, but telomerase does, because telomerase, that RNA template that is pre-bound to the enzyme, is complementary to those telomere repeats, recognizes the free ends of linear chromosomes, and then we'll use the pre-bound RNA template to extend the telomeres by partial complementarity, a couple of bases shown here, and then it's DNA polymerase activity to extend the telomere from the 5' direction to the 3' direction. Therefore, telomerase, through its enzyme activity and its RNA-dependent DNA polymerase activity, offsets the end replication problem that accompanies linear chromosomes. Telomerase is not in all of our cells all of the time. It's important during development. Stem cells and other types of things will have these telomerases, but when you start talking about adult tissues, the telomerase activity goes away. If you take a punch of your skin cells, fibroblasts and other things, keratinocytes that would ordinarily proliferate in a dish, they don't have high levels of telomerase ordinarily. If you try to culture them out on a dish, they'll grow. You give them a bunch of nutrients and other things to make them happy. They'll proliferate for a while, proliferate, and then they just stop. And what's happening is each round of those proliferation rounds, DNA synthesis needs to occur. There isn't telomerase to be able to maintain the ends, and the telomeres get shorter and shorter and shorter. And you can in fact age cells according to the length of the telomeres on them. Different species have different lengths of telomeres, but they all fade with age. And the reason why they fade with age is because telomerase doesn't persist in all cells forever, and as one cell replicates over and over again, you're always getting this whittling down. It's called telomere erosion. Pretty self-explanatory. When the telomeres have eroded to a critical point, certainly if the telomere is completely eroded away, I'm not sure how much left of the telomere needs to be there to trigger this change in cell behavior, but the cells receive a signal from those whittled down ends that cause it to undergo a cell fate called senescence. Senescence is descriptive in that in many ways it's exactly what it sounds. It's like cellular old age. Those are the cells, the keratinocytes, the fibroblasts in a dish that proliferate for a while, proliferate for a while, proliferate for a while, and then stop. They don't die. It's not apoptosis. It's not necrosis. They are metabolically active. You sit down on the plate, they'll be there, but they won't proliferate anymore. No more DNA replication, and that's the senescent cell fate. There's a couple of metabolic markers that characterize senescent cells. Almost everybody in the room, I can point to senescent, or you can think of senescent cells on your body. Moles, nevi, these are hyperproliferative lesions on your skin. They grow, they appear for a while, they grow, and then they stop. Why do they stop? Because those melanocytes senesce. They undergo a couple of proliferative doublings. They don't have enough telomerase to go any further, and then they stop. Benign lesions, nevi, senescent cells in our body. One other thing I should mention is that note because of the telomere repeat and that DNA template that's on telomerase, the cartoon here illustrates 1 or 2 cycles, but it's iterative. If telomerase activity is high or cells are engineered to overexpress genetically engineered telomerase that we introduced into the cells, they can continue elongating that telomere many cycles out. If you want to take keratinocytes or fibroblasts and immortalize them in culture, engineer them to express telomerase, and then you can get them to proliferate a lot longer than if you just take the X-plant out and grow it in a dish. All right, so that was that final hurdle for linear chromosomes. We're now ready for the rest of our time today to go back to the biowarfare agents. Anthrax. I mentioned this briefly in the beginning. Let's talk about its pathogenesis in a bit more detail. As I said, the lethal form is the inhaled form. When spores of anthrax are aerosolized and land in the airways of people, cell type that engulfs them are professional phagocytes, macrophages. We have resident macrophages in our lung tissue. They're surveying that epithelium, looking for all sorts of things that we might inhale. Other types of bacteria, dusts, pollution, all those things. Go in, scoop it up, phagocytose it. That process of phagocytosis, these macrophages are also very motile. So they phagocytose, and then they're patrolling in and out of the mucosal layer and into the tissue. Once that spore goes into a phagocyte of a macrophage, now those conditions are more hospitable for it to germinate and begin proliferating again. This is what it does. The bacterium proliferates, will in fact rupture that phagocyte, proliferate intracellularly in the macrophage, and then lyse the macrophage because it proliferates so extensively. And this is the onset of that fulminant infection that is universally lethal for the reasons that I described last time. We tie in the anthrax infections with this particular antimicrobial that's effective against it. And this will tie us back into DNA replication. Before doing so, we should begin with a little bit broader treatment of antimicrobial therapies for those with pre-medical inclinations or just general clinical interest while I take this class. So I'm going to talk about antibiotics and antimicrobials. These are chemical compounds, drugs used to treat infections. Antibiotics, which is used pretty generally, but formally refers to compounds that inhibit the growth of a microbe or a microorganism, but that are, if you will, naturally derived from another microorganism, soil dwelling in the tropics. It can be anywhere, but antibiotics are formally restricted to the biowarfare that microbes fight against one another. And they do it a lot, everywhere. In contrast, antimicrobials is a superset that includes antibiotics, but will also include small molecules and other synthetic chemicals that we have invented or discovered by screening and chemical synthesis to be able to perturb core processes in one microbe or another. I should also say that the term microbe encompasses all infectious agents that will set up shop in us. We're talking about bacteria today, but fungi are also microbes. Viruses also microbes. So that's a catch-all for anything that will infect us. All right, so antimicrobials. Some of the properties that would be desirable for you to use it as a medicine, all right, to prescribe it. Some of these are specific to the class of antimicrobials. Others are just generic to medicines. Right. If you want to kill something, like the microbe, you'd like it to kill the microbe and only that, not anything related to the host. So this notion of selective toxicity towards a life form is crucial for antimicrobials. All drugs, it'd be great if they were safe, easy, cheap to administer. Here's one that I want to speak a little bit more about because the distinction may not be immediately clear to you. Good antimicrobials should be bactericidal, meaning that they kill the if you're talking about for bacteria, or microbicidal, they kill the microbe, kill the bacterium, not bacteristatic, which simply means stopping it from growing. And this distinction can get blurred the way that often people will study a mechanism of action or start on antimicrobials. They'll start with a small inoculum in a tube, and they put in 1,000 bacteria, and then they let it go for 3 days, and then they see how many bacteria are at the end. A good bactericidal antimicrobial should have 0 bacteria, not 1,000 at the end. And the reason why it gets confounded is because if it's not effective at all, those 1,000 may be 10 million bacteria at the end of that time course. And so 10 million versus 1,000 looks like a big difference. But the problem is the 1,000 that hangs around is hanging around, finding ways to become resistant to that antimicrobial, which is the reason why killing and not stopping is the goal. And that's what I just said in point 4, right? So you don't want to find any grounds for it to become resistant, like it stopping proliferation rather than dying. Tissue distribution is important. If you have a gut infection, it's fine to take something orally if it's not absorbed very well. If you have a urinary tract infection, that probably wouldn't be so good to take an oral antibiotic that doesn't get absorbed by your gut. People are familiar with allergies. The last thing that to say a bit more about is this topic of activity spectrum. If one has a antimicrobial or antibiotic, what does it act upon? There are two broad terms that are, or two overarching terms that are thrown around: narrow spectrum antibiotics versus broad spectrum antibiotics. This speaks to the breadth of microbes that will be adversely impacted, that will be killed by that antimicrobial or antibiotic. Narrow spectrum would refer, let's see, things you've heard about. Gram-positive versus gram-negative bacteria. That's a key distinction. There are certain antibiotics, antimicrobials that work for one category versus another. So that will narrow things. Antifungals versus antibacterials versus antivirals. All of these things can narrow the spectrum. If one's infection is very clearly defined and there's a potent bactericidal narrow spectrum antibiotic, that's preferred. The danger of the broad spectrum antibiotics is that they do a lot of collateral damage on us even if we don't recognize it, because we have pounds of bacteria on us. Sorry to gross you out, but we do. All right, so a lot of us, we have more commensal bacteria by cell number on us than cells in us. And all of those in a broad spectrum antibiotic are going to get disrupted. And this opens up this notion of superinfection, where, what is superinfection? This would be a secondary infection that arises from an individual that got sick from another bug, but as a result of getting treated very aggressively with broad spectrum antibiotics, the collateral damage on the host commensal microbiome creates an opportunity for a much more dangerous bug to come into that sterile territory and set up a drug-resistant infection. What's the analogy? If you have a forest and there are all sorts of animals in there, top of the food chain is something like the predators. If those get wiped out, what will happen to the forest? The deer and the rabbits eat all the greenery, right? And the whole thing goes barren. Same things can happen. What's one you might have heard of? C. difficile infections. These are in the gut. Very dangerous antibiotic-resistant bacterium that only arises really in patients that are aggressively getting treated in the hospital with antibiotics for other infections, either systemic or gastrointestinal infections. There are many different ways that one can identify or design, target a route for developing an antimicrobial. This used to have a bunch of blue terms on it. I don't need to know all these, but I just want to give you a sense of what the categories are. How can you get specificity for microbes over us? One could target something like the cell wall of the bacteria. We don't have cell walls. And so you can, something like penicillin, classic example of an inhibitor of cell wall biosynthesis of bacteria. And the bacterium can't make the cell wall, can't divide, dies. There are others for antifungals that can disrupt specific functions of the cell membrane that are characteristic of that phyla. As we go into these lower classes, protein synthesis inhibitors, I'm treading lightly here because we haven't done protein synthesis properly yet. We haven't done metabolism either. Where we're getting to are these nucleic acid inhibitors. Each one of these, to just do that in a very broad sense, can have unwanted toxicities associated with it because we need to use things like metabolism. We need to translate proteins. We need to replicate a DNA. So there has to be some additional specificity determinant beyond simply those broad essential biological functions. One needs to target an enzyme or a process that is selective or specific for the microbe you're trying to target. This is the case for the antimicrobial and anti-inhalation anthrax agent called Cipro. Very powerful antimicrobial. You can tell it's an antimicrobial because nothing like this is made in nature. If you see a natural compound, look up penicillin, you'll see a big branched structure, a lot more complicated, but none of these exact functional groups. Definitely not a fluorine atom in there, for example. In the small n of people that have been have suffered from inhalation anthrax, it's the only known effective antimicrobial, and it falls into a broader class of antimicrobials called Fluoroquinolones, and it relates to the functional groups and the chemical structure shown on right. What Fluoroquinolones do is they inhibit an enzyme we learned about in last class, DNA gyrase, and not any old DNA gyrase, but specifically the bacterial DNA gyrase that reduces the supercoiling that is intrinsic to replication fork, movement, and DNA replication. We didn't show the structural parallels for DNA gyrases, but similarly to origin binding proteins and similarly to helicases, there's a lot more divergence in these enzymes from the prokaryotic to eukaryotic realms than you might think, given how important DNA replication is as a core biological process. So that's why it's a good target for these synthetic compounds. So we know replication now. We just got to take a quiz on it, right? So what's going to happen? Well, replication is going to proceed, but it's going to proceed up until the bacterium, the Bacillus anthracis, runs into this big rope tangle, like I showed in my demo a couple of classes ago. Overcoiled DNA, overcoiled, and then eventually this will either collapse or it will break. And so if the supercoiling is not resolved, the helicases will continue to push forward and is enough to give rise to breakage of the DNA. And more to say on that in another setting. So this is how Cipro impacts DNA replication, specifically in prokaryotes, specifically in anthrax, to be able to serve as an antimicrobial. Let's move on to mustard gas. To remind, this is a chemical agent that is an alkylating agent, a blistering agent, or vesicant. I looked this up periodically. It's not known really why all the blistering occurs, but the chemical modifications that are well documented as a result of mustard gas exposure appear to impact the keratinocytes. I already talked about those if you take a skin punch. They're especially susceptible to the chemical modifications and adverse effects, and that gives rise to the blistering that I described last time. That chemical modification of mustard gas occurs on DNA. Talk about DNA replication, and it refers specifically to a chemical modification on the base guanine. Chemical structure, mustard gas on the left-hand side, purine base of guanine on the right-hand side. I spoke before about how this chloride is a good leaving group on mustard gas for chemical attack. This nitrogen on the heterocyclic amine here is a good nucleophile to come in, attack here, chloride goes away, and now you have a chemical modification on that base, sometimes called an adduct. This means something stuck on there. And mustard gas has two of these chloride leaving groups. Double strands are pretty close to one another. Two guanines could conceivably be close to one another, and you still have another one of those modifications that can occur. And so if there are two adjacent guanines in the sequence, either on the same strand or on opposite strands, now you have a covalent cross-link between those two. Same type of chemical modification. Those cross-links can occur within the same strand if you go GG. They can occur between strands, so that's an intra-strand cross-link. between strands interstrand crosslinks, which would be GC. So the reverse complement of the C is G. That's why it goes off diagonal like that. Or they could just be a single modification. The other chloride group is left out there hanging. That's an alkylation event. I described that the last time, and that's just a single adduct, like I talked about before. These create a variety of problems. The polymerases, DNA polymerases, when they see those chemically modified bases, those bases as a result of the chemical modification are either conformationally constrained or chemically changed such that they no longer undergo proper base pairing. That's going to cause polymerase stalling or misincorporation, depending upon what the lesion is. For this, it's mostly stalling. Same thing occurs with the alkylation events. You have this big, comparatively bulky group hanging off the guanine. It's not going to base pair properly anymore. The really bad ones are the interstrand crosslinks, because now you've stapled the two parent strands together, and helicase still wants to plow through and unwind them. When you have these modifications and then the helicase activity persisting through the replication fork, you'll get this thing called replication fork collapse, meaning the whole thing breaks and falls apart. And the outcome of that is a double-strand DNA break. Both of the two pieces break in two. Very bad. That's the outcome of the bacterial DNA gyrase inhibition for Cipro. So all of these things, if they're extensive enough, and it's because mustard gas is so effective at the chemistry that it does, will be lethal to the cell, and if enough cells are affected by it, lethal to the organism. So talked about stalled replication forks, fork collapse, strand breaks. And yet, as a chemotherapy, mustard gas was one of the earliest ones that we used, or derivatives of it. And in fact, if you look for treatments for multiple myeloma, non-Hodgkin's lymphoma, chronic leukocytic leukemia, you'll see a drug called bendamustine. Where do you think the must comes from? It's this mustard gas, this nitrogen mustard group here, that does exactly the same thing on hyperproliferating transformed cells of those cancers. What's it trying to do? What's the rationale? Okay, and so where would you get some, yes, why might the cancer cells be especially susceptible to it? Saw a hand down here. All cells will be susceptible to it with regard to the chemical modifications. There's always the danger of that happening. What is, I mean, we haven't done the cancer module yet, but a characteristic of cancer cells that's different from normal cells. Yes, exactly. And so the unfettered replication that accompanies the unfettered proliferation of cancer cells makes them more susceptible to these DNA damaging agents. And other chemotherapeutics work exactly the same way. This is the reason why the side effects, hair falling out, gastrointestinal distress of chemotherapeutics, including bendamustine, these are also rapidly proliferating tissues that if you have these DNA lesions occurring, they can cause adverse side effects. So the oncologists seek to thread this window to give just enough for therapeutic efficacy, but not so much to kill the patient from the therapy. But early on, it was up in the air. All right. The modifications of mustard gas open up us to a whole general category of DNA damaging events that occur every day. If you are out in the sun, 25,000 lesions to your genome per hour that occurs. So wear your sunscreen. The first chemical modification that I want to talk about is a depurination event. We talked about purine modification with mustard gas, you can have certain carcinogens or metabolic stresses just take off the entire base. Guanine, hydrolyzed, and then gone. Now you just have a free sugar there. You've lost the information that's heritable. So this is one example of naturally occurring base damage. Another is that you can lose a chemical group on the base itself. One example of that is deamination. You have a cytosine here. You see the amine, which is crucial for the base pairing, for C to go to G. That amine can get removed. Now that converts cytosine to a uracil. U is going to pair with T rather than G. So that was going to cause a change. Adenine can also be deaminated, and it becomes another base called inosine that's recognized as I always forget this, as read as a guanine base. Another type of base change that you can have. You've seen this before I came here. Somatic hypermutation, B cells, affinity maturing, as they change around the positions. You probably didn't hear the gory details on this, but there's an adenine to inosine deaminase that B cells express and upregulate during the process of somatic hypermutation to incur these types of deamination events in the B cell receptor locus to experiment and evaluate whether there are higher affinity configurations of that VDJ recombination event that you learned about last quarter. As you might imagine, these need to be very tightly regulated because they regulate on where those deamination events occur and make sure they only happen in the cell types where they would be evolutionarily beneficial. Here's the major source of DNA damage if you're out in the sun. UV light has energy in those ultraviolet rays, and that energy can promote the formation of thymine dimers, two adjacent thymines giving rise to an intra-strand crosslink, as shown here, another form of DNA damage. Why are we all not just littered with hyperproliferative lesions and everything else? To get this far, we have evolved a very elaborate, we and all organisms on this earth, because we're getting battered with cosmic rays. We have radioisotopes in our bodies as we speak right now that are causing DNA damage. Evolving in time with all of these genomic insults is an extensive set of repair machinery to offset those damage events. Speak about one first, which is the base excision repair machinery, or BER. That will repair all of the events that I talked about before: thymine dimers, deamination events, depurination events. Do this at a very broad strokes level. There's a single base or a multi-base lesion here that's been modified. Base pairing has been disrupted in that area now as a result of the chemical modification. Now the double helix doesn't wrap quite like it ordinarily should. And the thinking is that distortion is a recognition for what are called repair endonucleases. I talked about endonucleases just as a blue term before. Now you're going to see them in practice here. Endonucleases cut inside a DNA sequence. And here they cut in the vicinity of where that chemical modification occurred. They don't fix the modification. They take out the entire sequence where the modification took place. So that's why it's called a base excision. It doesn't fix the base that got modified, it just kicks it out. Now there's a nick. The nick here will get filled in by polymerase. Sometimes other types of polymerases that we haven't talked about. Pol delta can fill this in, but there's also a polymerase epsilon or a polymerase beta, and these will come into play depending on how big the lesion is that needs to be filled in. But the filling in portion is exactly like we had talked about before. It will go in, runs right up until it hits the other end of the lesion, and then DNA ligase comes in and repairs the nick, just like the Okazaki fragments and everything else like we had described before. Very efficient, works fine if the cells are not ordinarily proliferating, if they're just not replicating DNA at the time. You have the genome there, recognize the lesion, cut it out, fill it in, and repair it. A much more dangerous DNA damage event is when there are double-strand breaks. These, in contrast to the chemical modifications, where can you get these from? Replication fork collapse, stalled replication forks, which means like a really gross insult, inter-strand cross-link. This can also occur, you usually see the lightning bolt. Doesn't mean your genome gets struck by lightning. It means it usually refers to ionizing radiation. All right, so if you go for an X-ray, this will happen. This is the reason why they limit your dose. Nuclear disasters, cosmic rays, things like that. These have enough energy to just go right through both double strands and create a break. Double strand breaks, because of their severity, we in fact have two mechanisms for repairing them. I'll talk about the first clumsy yet essential way of doing it, and then a more elegant one that exploits the diploid nature of our genomes. First, the clumsy one. It's really important, so I shouldn't knock it out that much, but it's called non-homologous end joining, or NHEJ. And the way that NHEJ works is that as soon as there's a double strand break, you have two free ends that are not telomeres, all right, in the genome. That creates a trigger for a class of proteins, which I'll spare you the details are, but there are end-recognizing proteins that go and bind and accumulate and assemble on those free ends to protect them and to try to repair them in such a way that they can be compatible with one another, the two free ends, that they can be compatible with one another to paste back together via a ligation event. And the compatibility there relates to having just enough of an overhang between the two pieces so that there's enough complementarity for the ligation, two ligations to occur on each strand. You saw this too. BDJ recombination, remember that imprecise joining and the plus or minus of some of the bases? That's non-homologous end joining trying to reconcile the free ends of the VD and the DJ segments. This is how you get junctional diversity. This is also how you spare your genome if there's a double strand break and you can't repair it by any other mechanism. If this is in a non-coding region of the genome, where if it's a regulatory region or it's a gene desert, usually it doesn't matter if you add a couple of bases here or there. You just paste it together, and at least you've preserved the broad strokes of what the chromosome looked like before the double strand break occurred. However, if this were to occur in the middle of a gene, addition of one or two bases is actually pretty catastrophic. That's probably unclear to you why at this moment, but when we talk about protein translation, I'll try to remember to come back to why. So we have another mechanism of DNA repair that, as I said before, takes advantage of the fact that we have two copies of each one of those chromosomes, and it's generally unlikely that both will break in the same place. We got one of those copies from mom, one from dad, right? If a double strand break occurs in one of those chromosomes, what takes place inside the cell is what's called a homology search. All right, so there's you have the free ends. There's a much more extended resection only on one of the strands, on each one of the strands where each one of the pieces where the double strand break occur. As those get whittled back in that way, the other free end looks for a complementary DNA sequence somewhere in the nucleus, basically fishing for the other chromosome. If mom's got broken, then it's looking for dad's chromosome. There are differences between those two chromosomes, but not so great that there's not the possibility of finding enough complementarity to bind to and hybridize to the other chromosome. When that homology has been identified, now it uses the other chromosome as a template, the polymerases, to fill in the gap. The same thing happens on the other strand. There's a strand crossover here that's not shown in this cartoon. And at the end, what results is, and then there's a ligation event at the end to bring the things together. Now what you have is a filled-in end that is informed by the genetic information in the other chromosome that has complementarity to it. To summarize this briefly, here you have no template. It's just cutting and pasting in a way to get it to paste together. Here, this is a template-mediated repair guided by the other chromosome that doesn't have the double-strand break. Questions on that? Because I need to build upon this in the next thing. Okay. Those processes of non-homologous end joining and homologous recombination can be exploited to engineer genomes. Have people heard at all about CRISPR? Follow the science times, something technological. Okay. CRISPR and this endonuclease called Cas9 exploit our double-strand DNA break machinery to perform genome editing. I'm going to walk you through this. So what is CRISPR? What is Cas9? Cas9 is an endonuclease. It in fact has two endonuclease domains. So this big gray thing is the cartoon bubble diagram of Cas9. The business end, its enzymatic activity, is conferred by these two endonucleases that cut DNA in the middle. That character in and of itself is unremarkable. I already talked about other endonucleases. You can cut DNA. That's fine. Bacteria can cut DNA, and you can cut DNA. Cas9 is from a particular order of species called Archaea. These are the things that live in like the deep sea vents, and sulfur pools, and things like that. And why Cas9 and the Cas9 family of enzymes are so remarkable is that the activity and localization of where those endonuclease activities become active is guided by a piece of RNA that binds to the endonuclease. Actually, formally two pieces of RNA, and that's this dual RNA strand-guided RNA shown here. All of the stuff in the black here is parts of the RNA that are important so that the RNA stays bound to Cas9. So it's like a lock and key type of mechanism that allows the RNA to plug in to the Cas9 so that they stay stuck together. Where the specificity comes into play is with this portion of the RNA called the CRISPR RNA. If I tell you that CRISPR stands for clustered regularly interspersed palindromic repeats, I don't think I've told you anything. So it's not a blue term up there. The origin of that name comes from where this was originally discovered. So Cas9 and CRISPR's RNAs are a form of adaptive immunity in bacteria. And so will win the Nobel Prize eventually. It's sort of a matter of a couple of years, all right? And the way that it operates is that when bacteria can get infected by viruses, a special category of viruses called phages. If they survive that phage attack, they will take pieces of that phage DNA, turn them into CRISPR clustered regularly interspersed palindromic repeats, and wedge them into their genomes. So that the next time if they see that phage again, they mobilize the Cas9 machinery, mobilize the CRISPR RNA from the past phage event, and then Cas9 goes around and looks for DNA that appears like that phase and will activate the endonuclease activity to try to chop up the phage virus. So this is how it arose evolutionarily. How are biotech companies and maybe one day pharmaceutical companies, and definitely right now in this building, labs, using Cas9 technology? We're using it for scientific purposes to change around DNA to our liking. And the way that we can do that is, well, we have the sequence for Cas9. We can specify whatever sequence that we like in this CRISPR portion of the RNA. And we don't have to target a phage. We can target the genome of the cell that's expressing the Cas9. The requirements for the targeting are very light. There's this little thing called a PAM sequence. You see it's blue here just so I don't have to recolor the image. Okay. It's a short little sequence that's absolutely required for the Cas9 endonuclease activity to be activated, but we have those all over our genomes. The rest in the red, the protospacer, that gives enough sequence information so we can target the Cas9 activity to one and only one location in our genomes. So we take cells, engineer them with a single guide RNA together with Cas9. Cas9 gets expressed in the cells. The single guide RNA gets expressed in the cells. They load up with one another. And then the Cas9 looks for a complementary sequence to the CRISPR RNA sequence. And once it's found, as shown here, by base pairing in the genome, when there's proper base pairing and you have that protospacer adjacent motif sequence, the two endonuclease activities get activated, double cut right there. Now you have a double strand break in one location in the formerly two locations in the genome because you try to target both of them. Okay, the two chromosomes. If you just do that, what will occur overwhelmingly in the cells is non-homologous end joining. So you'll have the cut, sends off a DNA damage response, all those end binding proteins and other things, we'll try to find them and paste them back together. The net result of that ordinarily is a couple of random insertions or deletions that's shown there in the green that have now altered the genetic sequence of that organism in a heritable manner right at the place where you wanted to. And so I talked about how a few bases added or deleted in a coding region of a gene, and a gene where the gene is actually expressed, can be very deleterious. This is how we make knockouts now. I think you had a slide talking about knockout technology, being able to delete the protein expressed by changing the gene sequence. We now do this on demand pretty much in any cell that we want to by using CRISPR-Cas9 and non-homologous end joining to disrupt the way in which a protein is expressed in cells. And if we target both of the alleles of the gene and the deletion of that gene does not cause the cells to die, we can make knockouts of any gene of interest. So that's pretty cool, pretty useful in the lab, pretty beneficial if people are using this for, there are certain diseases that have deleterious alleles that would, sickle cell, the sickle allele, if you could knock that out, patients cured. And actually they're using this. There are patients that have had bone marrow transplants with CRISPR-edited hematopoietic stem cells by this technology, just using the non-homologous end joining machinery. The part on the right-hand side, which is a bit more elaborate, invokes homologous recombination. And so the way that this works is you do the same thing, CRISPR-Cas9, cutting, open it up. But in addition, you provide the cells with something that looks like the other chromosome. So this is another piece of DNA that if you're engineering cells, you just get another piece of DNA in there. The red looks like the red here. The red, the blue, and the black looks like the blue here. But inside, there's a new piece of sequence, which is what you want to put directly in between the gap that you just created. Why would you want to do this? If you have a broken gene or protein that you want to try to fix, so there's a sequence that you'd like to cut out and replace with something else, this is the way you would need to do it. If you wanted to make a protein that, by the time it's translated, has a fluorescent protein tagged to it every single time, the green in there you could put in the sequence for a green fluorescent protein, and then the protein gets translated, and then it translates the green protein right in frame with it. Everything is endogenous, no overexpressions or anything else from there. People do this routinely now also. Much lower percentages that this succeeds with, but you can get it to work in the lab. And so by exploiting some of these repair processes, biotechnologically, scientifically, with some basic fundamental discoveries in deep sea vents and sulfur springs, we now have really powerful tools to be able to study gene function and then hopefully one day repair genomes for human therapy. If you find some of these things cool, I should update this slide because Joyce Hamlin has retired and is totally out of commission, but Aninja is our DNA replication guru in the Department of Biochemistry and Molecular Genetics. And I need to update the slide because there's in fact a lot of people in that department doing really cool things with CRISPR-Cas9. So if you're interested in those, you can look them up. Any questions in the time we have remaining? All right. Good luck on any final edits to your peer review, and I'll see you on Thursday.